students. So today we are going to have a practical on green fluorescent protein, which is GFP, and its transfection in cells. So let us discuss what green fluorescent protein is. Basically, this protein has a molecular mass of 26.8 kilodaltons. It was first purified from a jellyfish named as Aquaria victoria in early 1960s by a Japanese scientist, Osamu Shimomura, who received the Nobel Prize in Chemistry for his discovery in the year 2008. This particular protein is 239 amino acids long, consisting of a beta barrel-like structure with a chromophore at center, and widely used as a reporter protein in various assays and tracking experiments. Say, suppose you want to overexpress a gene and you want to also see whether the gene is essentially getting overexpressed or not. So how do you want to uh, check whether the transaction has occurred or not? So for that, you need a reporter molecule, which is cloned in frame with that particular gene. So as and when the gene expresses, the reporter molecule would also express, and when excited with a particular fluorescent excitation signal, it will emit fluorescence at some particular emission wavelength. So this is mostly important in transfection experiments as a reporter signal, as I just said. Now, if you look at the structure, the structure is displayed here, and you can see there are beta barrels, beta strands consisting of a barrel, and in the center you have a chromophore which consists of three amino acids, which essentially constitute the chromophore, being serine, tyrosine, and glycine, S, Y, and G, as the one letter symbols would be for the amino acids. And when excited at a particular excitation wavelength, it will emit fluorescence at a particular emission wavelength, which is then captured using a fluorescent microscope. So that's essentially what GFP is. Now let us discuss the basics of transfection technique. So transfection is a term which is specifically applied for mammalian cells, and it involves the introduction of foreign genetic material, could be DNA, RNA, into mammalian cells. Now, there are two kinds of transfections which are performed, one being stable and the other one being transient. So let us discuss what transient transfection is. Transient, as the name suggests, this transfection is short-lived, and the foreign DNA here will not get integrated into the host genome. So say suppose you perform a transfection and you want to observe the effects of that transfection. First of all, in a transient transfection, those effects would be short-lived, and after some subcultures or several cell divisions which would occur, this particular foreign nucleic acid would get digested, and the downstream effects of that particular nucleic acid or the DNA molecule would also fade away. So that's why it is being termed as transient transfection. Talking about stable transfection, so this has a long lasting effect, and the foreign DNA is integrated into the host genome. Usually, what is performed in a stable transfection is basically that while designing the construct, which is the plasmid that encodes the target gene which is to be transfected, apart from that, it also contains a selectable marker for antibiotic resistance. So that particular selectable selectable marker for antibiotic resistance would confer antibiotic resistance only in the transfected cells. So over generations, when you want to apply a selection pressure, wherein you treat these transfected cells with a dosage of particular dosage of antibiotic that essentially doesn't kill the cells, you would observe that with subsequent cell divisions, only the transfected cells would finally remain. And we would be left with a very small population of cells which contain the transfected DNA, because we had applied the selection pressure and now we have just arrived at our transfected cells. So that's how we select our transfected cells. And in this case, the foreign DNA gets integrated into the host genome over a period of time. Hence, it is being termed as a stable transfection. Now the question comes, why should we transfect? So the reasons could be numerous. So I'm sticking here only to two reasons. First of all, if you want to overexpress a gene or say a microRNA, and you have experiments wherein you want to study the effect of this particular gene on some targets, or the effect of a microRNA on its gene targets. So you want to overexpress them in a cell. So that's why you want to do a transfection. The second reason could be for knocking down a particular gene. So for that, you require certain molecules termed as siRNAs and shRNA. So these molecules would essentially block the function of that particular gene. So this way you can expect to have a knockdown of the gene and then compare the particular effect of the knockdown to the control, wherein there is no knockdown occurring. Let us discuss different transfection methods. So I have listed out three transfection methods here, which is cationic lipid mediated transfection. So here in this, you have a nucleic acid, DNA, suppose, that we all know is negatively charged. And you have cations in the reagents that these companies manufacture. So these essentially form complexes with each other. So the positive and negative charge will attract each other, and through electrostatic interactions, they would assemble into a complex. And then these complexes are delivered into the cells. So the typical uptake method here for the cells is through the endocytotic pathway. And that is how your foreign DNA gets introduced into the cells. Now, the second method is electroporation. So, this method involves uh, putting the nucleic acid and the cell into which you want to perform the transfection in a conductive solution that would conduct some kind of charge. Then you apply a short electrical pulse, which would essentially open the lipid bilayer and enable the uptake of the particular foreign DNA into the cells. So that's how electroporation works. Then you have in vivo methods when you use different vectors such as adenoviruses, retroviruses, lentiviruses, and adeno-associated viruses. One point to note here is that these viruses are engineered in a way that disables their deleterious functions on the hosts. So now you have disabled or disarmed viruses, and you use them as vectors for performing your transfection. These techniques are used wherein you want to have transfection in some very difficult to transfect cell types. Now, we all know that in cell culture, we deal with many kinds of cell lines. Some of them are very easier to transfect, whereas some of them are very difficult to transfect. So, a justifiable choice here would actually depend on the first of all the kind of cells that you're using, whether they're easier to transfect or not. Then you would also want to see whether that particular transfection method is cytotoxic for the cells. Then you would also want to look at whether the transfection would work well or not. So that we call as transfection efficiency. That's a term we used in transfection commonly. So that's how we arrive at these transfection methods and finally make a choice. So let us discuss the lipid-based transfection method. Now, this method is preferred due to the lower levels of cytotoxicity it would confer as compared to other techniques. Then, we prefer it because transfections performed using this method have a higher efficiency as compared to other methods. What is transfection efficiency? This is defined as the proportion of cells which express the transfected gene or nucleic acid. Now, this technique is also very versatile in the types of cells on which you want to use them. Say, suppose you want to perform a transfection in HeLa cells, or you want to perform transfection on U87Mg cells, or you want to perform transfection on breast cancer cell lines. So, this technique has been time tested to work on a variety of cell lines. That is why it has been used primarily in various research labs throughout the world. And in even our lab, we use this particular technique. And one of the most important reasons is that this technique is very quick and easier to perform. The protocol is also very straightforward, doesn't require much of uh, extra training or knowledge beforehand. We can simply refer to the protocol and perform a transfection. Now, this is essentially a flow of how this particular transfection works. And this is how lipofectamine, which is a reagent, transfection reagent, sold by Thermo Fisher Scientific, works. Now, you see, you have the foreign DNA here, and you have the cation lipid transfection reagent, which is supplied by the company. You apply it to the mix. You make a mix of DNA and this particular reagent, and we leave it for incubation for a certain amount of time. This incubation would allow interaction of the negatively charged DNA with these positively charged cationic complexes, and it would lead to formation of a DNA cationic lipid complex. Once this complex is assembled, we finally incubate this particular mix, and this is followed by putting this particular mix on cells which are already present in a cell culture plate. And though this is lipid based transfection, and we all know the cell membrane also consists of a phospholipid bilayer, owing to the similarity in the terms of the chemical makeup of these particular lipid moieties in the transfection reagent and the phospholipid bilayer, they would recognize each other. And this is how, through the endocytotic pathway, the cell would quickly take up the foreign DNA, and you will have expression if you have successful transfection. Let us discuss about lipofectamine transfection reagent. Now, within this particular category of lipofectamine transfection reagent, which pertains to the class of lipid-based cationic complex forming transfection reagents, there are various subclasses. So you would want to actually appreciate here how many kinds of lipofectamines are manufactured and sold by Thermo Fisher Scientific as a company if you want to choose lipofectamine as a transfection reagent. So this choice of having a particular transfection reagent is dictated by various factors. Most importantly, the kind of cells you're working with, whether these are primary cells or they are stem cells, or they are cells as special as neurons. And then it also depends on the payload you want to deliver. For example, you want to work with DNA, you want to transfect a microRNA, you want to transfect shRNA or siRNA. So then you can make a choice based on these particular factors and arrive at a transfection reagent that best suits your needs. So you have lipofectamine 3000, 2000, RNA max, messenger max, CRISPR max, and stem. Now, one of the most common ones are lipofectamine 3000, 2000. Now, what we use primarily in our lab for research purposes is lipofectamine 2000. And there are various reasons for using lipofectamine 2000 in our scenario. So we want to have a versatile reagent, first of all, because if we are working with, say, suppose 3 different or 4 different kinds of cell lines, we might want to choose one particular transfection reagent which would essentially work for all cell lines. Then the kind of payload we want to deliver, and say, suppose you want to ensure that your transfection is going nice in terms of the transfection efficiency, which essentially has to be optimized. So no transfection comes very straightforward as per the protocol. Once you sit on the bench in the lab, you'll have to optimize the transfection based on the number of cells initially you want to seed in your cell culture plate, followed by the amount of DNA or the nucleic acid you want to take for the transfection, or the dilution you want to make of this particular nucleic acid to the um, to the to the complex. So that also plays an important role here. And then what exactly is the workflow you want to follow in terms of incubating the cells once the transfection is performed and changing their media, which would be discussed in the upcoming slides. Let's discuss about lipofectamine 2000 as a transfection reagent. So there are various reasons why we prefer to use lipofectamine 2000 for our transfection purposes. First of all, it gives us superior transfection efficiency, as I had discussed previously about what transfection efficiency is. Then we find it to be very versatile and can be applied over a broad range of cell lines. So if you would note that in the previous slide, I have just discussed that the choice of transfection reagents is primarily dictated by the cells you are working on and also the payload that you want to deliver. Then we have higher levels of recombinant protein expression in our cells when we use lipofectamine 2000. And we can get high yields of protein and then finally harvest the proteins for downstream applications or performing Western blots and doing some other conformations or probably protein assays in the long run if you want to study the activity of those proteins in the cells. Then it follows a very simple protocol, as I just told that the protocol should be straightforward and must be simple for anyone to follow. Then it's having a superior performance when you want to have co-transfection, wherein you want to transfect a plasmid DNA as well as an siRNA along with it. So a co-transfection would essentially involve two transfections occurring at the same time in a cell, and wherein you want to establish a relationship between the two molecules being transfected, or probably transfecting something and then uh, kind of inhibiting it in an experimental setup. So that's where you want to use co-transfection. Finally, one of the reasons for using lipofectamine 2000 is that it is one of the most highly cited transfection reagent in literature so far. So if you read various research publications and you go to their materials and methodology section, most of the scientists have been using lipofectamine 2000, and this has been a very time tested reagent which is being used in cell culture techniques. So that's the logic behind using lipofectamine 2000 as a transfection reagent. So let us start discussing about the detailed protocol of lipofectamine 2000 DNA transfection reagent. So this works best for plasmid DNA. It will also work for various other molecules that you want to transfect, which could be shRNA, siRNA. But in those cases, the amount of the nucleic acid that you take would slightly vary from the amount of DNA which is mentioned in this particular protocol. So that calls for again optimizing your protocol while performing an experiment depending on the payload you want to have. So the timeline here shows on day zero that you have to plan your experiment. So planning involves first of all seeding your cells into either a 96 well, 24 well, or a 6 well plate. Once you're done with this, you'll wait for 24 hours and let the cells incubate and get attached to the culture dishes. Now, in order to have 70 to 90% confluence on the day of transfection, you must be sure as to how your cells grow in terms of, first of all, how much amount you have seeded. Secondly, the generation time of the cell line. That is a very important factor. Once this has been determined, you'll know beforehand that how many cells you have to seed. Typically, the amounts which are present here are very indicative. So we can seed close to the indicated amount of cells which have been mentioned here against 96, 24, and 6 well plate numbers, and arrive at 70 to 90% confluent cells. But again, cell line specific protocol also varies. So you'll find various cell line specific protocols for lipofectamine DNA transfection. So once your cells are ready, the very next day you let them, first of all, you let them incubate for 24 hours, and the very next day you start your transfection. For that, you take lipofectamine reagent and make 4 dilutions of that in OptiMEM medium. OptiMEM medium is special in terms of having a very low amount of serum as compared to your regular cell culture media, which essentially has approximately 10% of serum concentration. The logic behind using low concentration of serum is to enhance the chances of the uptake and primarily the formation of the lipid DNA complex, because it has been shown in studies that serum interferes with the formation of such complexes. So we use a reduced serum medium. There are special transfections which require complete absence of serum as well. So those have to be dealt with accordingly. So you make these 4 dilutions. Now the logic behind making 4 dilutions is because if you are performing a transfection for the first time, you don't know which concentration of lipofectamine reagent would work best for the cells you have and the culture dishes that you are taking, and also the payload that you have. So it's good to always work with 4 concentrations, and maybe through 1 or 2 experiments, once you have a practice, you'll arrive at a particular concentration of lipofectamine reagent that suits your needs in the specified conditions for a particular cell line. You have made these dilutions. Now you dilute DNA in OptiMA medium. You have diluted your DNA. There's a fixed amount of DNA which has been shown here as zero point 0.5 to 5 micrograms per microliter. If the concentration of the plasmid DNA is in this range, then you have to accordingly take that particular amount of DNA in terms of volume, in terms of microliters. So you dilute your DNA as well in OptiMA medium. Now, what we do is we add the diluted DNA to diluted lipofectamine reagent in a ratio of 1:1. Take equal amounts of the dilutions of lipofectamine to the particular dilution of DNA and simply mix them. Now comes the step where you want to have the formation of the lipid DNA complex. So you let it incubate for 5 minutes at room temperature. This 5-minute incubation would ensure that the complexes have been assembled before you apply them to the cells which are growing in a plate. Now, you take the DNA lipid complex and apply it to your cells which are growing. It's always good as a practice to change media of the cells before adding this particular mix into the cells. So that would ensure that you'll have, first of all, fresh media added to cells, and cells are in an actively dividing state, and the cells would keep on growing with the replenished media that you add just before transfection. Once this is achieved, you have to simply add your DNA lipid complex to the cells as per the volumes which have been mentioned again for 96, 24, and 6 well plate. And it has also indicated the final DNA amount which will be used per well in a plate. So it also indicates the final lipofectamine reagent which is being used per well in a plate. Once you have done this, you'll have to wait for the cells to take up the foreign DNA and probably wait for 1 or 3 days while incubating these cells, which have now been administered with your lipofectamine DNA mix for probably 1 or 2 days, and incubate them at 37 hours, 37 degrees Celsius, I'm sorry, and then you analyze your transfected cells. Also, one thing which has not been mentioned here as a good practice is after you have just performed your transfection, it's good to always supplement the cells with fresh media. But you should be very careful while doing that step, because if you do it too early, then there might be a chance that whatever foreign DNA that you have added to cells, that might also get removed while replenishing your media. So it's always good to wait for at least 5 to 6 hours before you decide to change media right after transfection. Now you don't have to disturb your cells. Once your media has been replenished, you'll have to incubate them at 37 degrees Celsius for some hours, and then you can constantly monitor the transfected cells. Now, if your cells have a good GFP signal, you'll get to see that under a fluorescent microscope. So that's pretty much about how lipofectamine transfection reagent protocol works. So this protocol might have variations, and these variations depend on the particular kind of cells that you're using. I mean to say the cell lines which you're working with, and it could also work differently for the payloads that you want to deliver. Say it is if it is a DNA, or if they are shRNA, siRNA, or microRNAs for that matter, you'll have to vary and play around with the protocol. So there are slight variations in those protocols, and a very comprehensive information about how the protocol has to be varied for different payloads and um, different across different cell lines has been mentioned on Thermo Fisher website. I'll provide you a link so that you can visit that particular page and kind of get an idea as to how this particular transfection can be carried out, which best suits your needs. Say now that you have transfected your cells and you want to see how they look. So here's a schematic which has again been taken from Thermo Fisher website, and they have displayed that in a 96 well plate for varying amounts of the reagent which were used per well, you have different transfection efficiencies. So the take home message from this slide is that not all cells would show similar transfection efficiency. As I have repeated many times, your transfection protocol would sometimes or most of the times vary depending upon the kind of cells that you are using, the cell lines to be specific here. So we can vary the amount of DNA, the reagent, lipofectamine reagent, and the dilutions that we had just discussed in the previous slide, and arrive at the best combination which suits us in a particular experimental scenario. So this requires some level of optimization. Once you are done with this experiment, you'll arrive at some kind of results, and based on those results, you'll want to optimize your transfection for subsequent experiments based on the cell lines that you're using. So if you see the particular schematic and the image that they have mentioned here, what they want to say is in some cell lines, the transfection efficiency is very good even with very low amount of lipofectamine reagent. So take the example of MCF7 cells. Look at how nice the GFP signal is being captured in that particular scenario. And then if you look at 3T3 cells, for example, and you see the highest amount of lipofectamine which has been added per well to the cells, you see that the transfection efficiency is not as great even with um, the highest amount of lipofectamine reagent which was added to the wells. So this wants to tell you or convey a message to you that the cells which you are working with are the primary determinants of how your transfection would work. And hence, it's always advisable to have cell line specific protocols for certain cell lines in which it has been proven that transfection could be really a difficult job to perform. So that comes by a thorough revisit of the previous literature, which has been reported in an experimental scenario for that particular cell line. And yes, cross referencing with the Thermo Fisher Scientific website, and finally arriving at the dilutions, the amount of DNA you want to use, the reagent you want to use, and the number of cells you want to seed based on the conditions and the experiment you wish to put up. So far, we have visited the detailed protocol, and we have also had a look at the expected results you might get upon transfecting GFP in cells. Now, in order to have a better look and understand it in a better way, let's go to a video which is available on YouTube. And this has been prepared by Thermo Fisher Scientific, and it's a protocol for a generalized lipofectamine transfection scheme. And through this video, you'll get to learn as to how a particular transfection experiment is set up, and it has briefly described within a couple of minutes as to how a transfection is to be performed, what conditions are to be maintained, and what are the good laboratory practices, most importantly, while working in a cell culture lab while you are setting up any experiment. So I'll request you to browse through this link and open this video, and please have a look at this entire video once properly. This will be very helpful for you. So what I'll do is I'll share the video link in the chat, and I'll request all of you to kindly open the video. It's just a 4-minute video you all can have a look at. And after that, we'll proceed to the next slide. Is it okay?
Yes, sir.
I hope you would have all received this video link in your chat.
Yes, sir, it's available.
Okay, please all of you go ahead watching this video and just let me know once all of you are done. I'll wait for 3-4 minutes.
Okay, sir.
Shall I proceed to the next slide now? I hope if everyone would have watched the video.
Yes, sir.
Yes, sir.
Sure, then I'll just play the next slide.
So far, we have discussed how transfection experiment has to be set up and how you might expect to have a signal. But it's very important to also know that how you will operate a fluorescent microscope. So it's important to know how to visualize your GFP molecule. You need a fluorescent microscope, you need transfected cells, and you need some knowledge about the fluorescent excitation and emission wavelengths for GFP. So the excitation wavelength is 395 nanometers and the emission wavelength is 509. You'll have to set your fluorescent microscope filters accordingly or manually put in these values if you are using a fluorescent microscope, a superior fluorescent microscope. The schematic shows you how a fluorescent microscope works, and you have a source of light, you have an excitation filter which is set as per your reporter protein or a molecule. You let that particular excited beam of light fall on your sample. The reporter molecule would get excited and emit in a particular wavelength. If you have a particular emission filter set at a desirable wavelength, you'll capture that emission and you'll detect it using an eyepiece, or maybe you can quantify as well the GFP signal, and that will help you in making calculations about the transfection efficiency and monitor it on a real-time basis. Say you want to have a transfection, and you're doing your fluorescent imaging maybe after 2 days after the transfection is done. You can have a look at as to how the transfection has progressed over a period of days. And since it's a transient transfection that we have been discussing about, you'll also get to see that after a certain amount of days, the signal would essentially fade.
Excuse me, sir. Sir, your audio is the voice is extremely low.
Oh, is it?
Yes, sir.
So was that happening for all the slides or just this slide?
No, just this one. Like after watching the video, when you
Right, right.
started explaining a few things.
Yeah, let me just check this. So I'll play that slide here and I'll explain it. I think that's better.
Yes, sir.
Yes, sir.
So first of all, am I audible?
Yes, sir.
Okay, so after watching the video so far, you would have understood how we can perform a transfection. And specifically, when you are dealing with GFP, which is a widely used reporter protein. Now, this slide basically talks about the experiment once has been done. So, what has to be done after that, which is visualization, of course. So, what all you require here it would be a fluorescent microscope, your cells, and some knowledge about your GFP. I mean, any reporter protein, GFP in our case. So the wavelengths which have been established here as the excitation wavelength for GFP would be 395 nanometer, and the emission peak would come at 509 nanometers. So this schematic, as you can see here, is about fluorescent microscope and how it works. Basically, it has a source of light and an excitation filter, which you set according to your excitation wavelength. That goes through a dichroic mirror, which would let it fall on the sample. The sample molecules would expressing GFP would get excited at this particular wavelength, and they would emit uh, light, which would reach the eyes of the one who's operating the instrument through an emission filter, which has been set, of course, at 509 nanometer in this case. So that's essentially what I wanted to convey using this slide. Coming to the next slide, this is about how to troubleshoot your transfection. Now, what I have shown you here throughout this lecture has been a very basic protocol, and there are various optimizations which have to be performed in order to have a good transfection efficiency, which we had just talked of in one of the slides. Now, this link would be available to all of you once I upload this particular lecture on MS Teams for your convenience. So you can have a look at this link later on perhaps. The major message that I wanted to convey using this slide was about how to perform transfection in cells, and I majorly focused on lipofectamine as a transfection reagent and why we use it and what all conditions have to be optimized. And we just went through one small video wherein we can have a look at how exactly these things are done, because without seeing a video we cannot perhaps appreciate how all the protocols are being followed. So this brings us to the end of lecture 1. Now I'll take doubts and questions from you, and then we'll proceed to lecture 2. Please go on. Please ask any questions if you have. Anyone who has a question, any doubt? Yeah, so is it fine if I close this lecture and we proceed to the second lecture now?
Yes, sir. No doubt for now.
Yes,
Sure.
sir.
So what
Yes,
I'll
sir.
do, I'll share both of these two lectures together in MS Teams. I'll upload these. Okay, so you can have a look at the video links as well as this troubleshooting link so far, which we haven't gone through here, but you can just have a look at how to troubleshoot your transaction, just in case someone wants to ask you about something which hasn't worked, and for your practical knowledge as well. So yeah, let me just close this video and I'll play the second presentation now. Yeah, so am I audible first of all?
Yes, sir.
Yes, sir.
Let me just share my
Yes, sir.
screen once again. Let me just share my screen once again. I hope it will not create any technical snags later on while this presentation is going on. Just give me a second. So I hope it's visible and I am audible as well. I'll just play the second lecture now. And if just in case you don't hear any sound, just let me know immediately. I'll correct it.
No, sir, you are audible.
Okay, okay. Just in case if you don't hear the presentation sound, just let me know.
Hello students. So today we are going to have a practical on cloning of a gene into an EGFP construct. So if you remember that we had previously discussed about transfection of GFP into cells, and then how GFP serves as a reporter molecule for any particular transfection or even localization of a particular protein in whichever cellular compartments as the case may be. That is why we are going to have a lecture today on this, and we are going to have an insight into designing primers and getting the gene as well as the GFP sequence cloned into the particular construct of our choice, and then assembling the construct and finally seeing it in totality as to how the fusion product would be formed. So before we go on designing primers, let us first understand some basics. We are all aware that the central dogma of molecular biology, it says that genetic information flows from a molecule of DNA through messenger RNA molecule, which finally gets translated into a protein, and then we does have a phenotype based on the genetic code or the parent DNA sequence. So here it has been shown that DNA has got two strands anti-parallel. One runs from the 5 prime to 3 prime direction, the other is from the 3 prime to 5 prime direction. Note that when we are not depicting any polarity of the particular strand and it's just a single strand given, by default it is assumed that this is the 5 prime to 3 prime strand from left to right running in that particular direction. So as you can see here, a DNA molecule has got two strands. Now, one of the strands is called a plus strand, and the second strand is called a minus strand. The plus strand is the one which is known as a coding strand or a sense strand, whereas the minus strand is the one which is known as the antisense or the non-coding strand of the particular DNA molecule. So now, if there is a transcription which is occurring from this particular DNA molecule, we must remember that the messenger RNA that would be formed is nothing but just a carbon copy of the plus strand. The only difference being that wherever thymines were present in that particular DNA, they would be replaced with uracil in this particular messenger RNA. Also, one important point to note here is that While messenger RNA is being transcribed from that particular DNA molecule, the antisense strand, the minus strand, it serves as a template strand, and here the extension would occur and it would lead to formation of this particular messenger RNA. So being a template strand, it means that this particular minus strand is perfectly complementary to your plus strand. And hence, when the primers would anneal and transcription would occur, you would have assembly of messenger RNA, which would be perfectly complementary to your antisense strand or a carbon copy of your plus strand. Just that it will have uracils instead of thymine as per the sequence of your plus strand. So that's very important because we have to sit and design primers, and we must know where would the forward primer get annealed, where would the reverse primer get annealed. So this piece of information is very necessary for now. This slide talks about complementary DNA. So what exactly is complementary DNA? Let us first understand that. We all know that in eukaryotic transcriptional machinery, we have the formation of messenger RNA, which consists of both coding, which are the exons, and non-coding regions that are the introns present in that particular messenger RNA. Then we have the splicing event which occurs, which leads to the removal of these intronic regions and leads to the formation of only the coding sequence of the messenger RNA after splicing, which consists of exons. Now, what we do in lab is, if we have cells and we want to isolate total RNA or messenger RNA from the cells, we first get the messenger RNA from the cells, and this messenger RNA, as we all know, has been formed after a splicing event. So we know that we don't have any introns in this, and we just have the coding sequence. Now, this particular messenger RNA is taken as a template, and a reverse transcription PCR is done to obtain the complementary DNA using reverse transcriptase as the enzyme. So, this complementary DNA, which is now being formed, consists only of the coding regions that are the exons and doesn't contain any introns. This is of paramount importance because if we want to perform any cloning, we must only have the coding sequence because we want finally that gene to express through the cellular machinery. So if we want to do that particular expression in bacterial systems, because we all know that bacterial systems do not consist of such splicing machinery, it is only the eukaryotic cells. And mostly in laboratories, for our ease, we use bacteria as a model system because of their various advantages that we all know. For example, they have quick doubling time, and the mean generation time is very less, and we can obtain large volumes of culture maybe for various experiments. Say, suppose you want to have protein in very large quantity. So that is the reason why we go ahead with these protocols. And now we are, so the message from this slide is that we have obtained the cDNA, which is exactly the coding region, and it doesn't contain any intervening non-coding sequences. For our PCR reaction, for which we have to design primers, this particular cDNA would serve as a template. Now that we have discussed about the template and the two strands of DNA in depth, let us first discuss about what forward and reverse primers are before we even go ahead designing primers. So in a PCR reaction, we need two primers, forward and reverse. What is a forward primer? A forward primer is nothing but the sequence of your plus strand being read in the 5' to 3' direction. So why am I saying this? You need to understand this. If you look at this particular figure, you are seeing that the forward primer is running in the 5' to 3' order, but this is complementary to the minus strand of the DNA. So we all know that the plus strand is perfectly complementary to the minus strand of the DNA. Hence, the forward primer is also complementary to the minus strand of the DNA because it is running in the 5' to 3' direction. So the forward primer is nothing but the sequence of the plus strand being read in the 5' to 3' order on that particular strand. But remember that primer length varies depending upon various conditions that we'll discuss in the upcoming slides. Ideally, any primer would have a length of 18 to 25 nucleotides. So we can say that if you're designing forward primer for any particular template, you can simply take the plus strand sequence and just take the initial 20-25 bases of that particular sequence, and hence you obtain the forward primer. Now coming to the reverse primer, as you can see in this figure, the reverse primer also runs in its 5' to 3' order, and hence it is complementary to the plus strand of the DNA. Now we all know that we write the primer, or even any for any DNA sequence, we write all the sequences in the 5' to 3' order by default without even specifying any polarity. So if you see For a plus strand, we all know that it starts with the start codon, which is ATG, and ends with the stop codons, which in this case here given is as TAA. So now if you want to design a reverse primer for this particular strand, you need to remember that it would be a reverse complement of the plus strand. So if the plus strand is ending as TAA in 5' to 3' order, your reverse primer sequence in 5' to 3' order would be its reverse complement. So it would be TTA and so on. So that's the basic message to learn from this slide. Just that you need to be very careful about the sequence of the reverse primer. Because if you want to design primers and then you want to order them, by mistake, if you have written the sequence of the reverse primer in the wrong polarity, the entire experiment would fail. So just be careful about the reverse primer sequence. So before we start designing primers, let us first understand the fusion construct that we have. Now, this is being called as a fusion construct is that the vector backbone here is of pcDNA 3.1, and it has got a sequence of eGFP, which is enhanced GFP, as a reporter, and you have your insert. Now, if you look carefully at the sequence, so it is under a CMV promoter, and then you see this MCS region wherein you have various, you have this MCS region wherein you have so many restriction sites available, and then you will have eGFP, which would lack the start codon. So let me just explain this quickly. If you have the MCS where you want to perform your cloning into this particular vector and which has got eGFP already, or say suppose you just have pcDNA 3.1 and you want to insert eGFP first in it, followed by the insertion of your particular gene of interest. So the procedure seems to be a bit complicated, but let us just try to understand this particular thing in brief. If you see this MCS, it consists of various restriction sites. So you can see various enzymes, NHE1, HND3, BAMH1, EcoR1. And then there's this eGFP sequence which lacks a start codon. Now, why is it lacking a start codon? The reason is that there is something which we call as cloning in frame. So if we take our particular gene of interest, say X is the name of that particular gene, and according to this construct, if you want to have the gene first followed by the eGFP sequence, you need to remove the start codon from eGFP. Why is it that essential? Because if you incorporate a start codon in eGFP, what will happen is upon translation, it will form two proteins instead of one hybrid fusion product because it will have two start codons. And one important point to note here is that you need to also remove the stop codon from your particular gene of interest because you'll have the start codon of that gene of interest and then you'll directly have eGFP beginning without its start codon. So now this entire DNA sequence, this would get translated into a fusion product and hence you will have the expression of eGFP as well as your gene. Now you simply take this entire fusion construct and say suppose you perform transfection in cells, you would see EGFP signal coming from the cells which have been transfected. So that's the goal of the particular experiment. And this is why this particular fusion construct was designed by an MSR student in our lab previously. And she has been very kind enough to share the sequence with us and the particular construct. So that's how we are going to perform our cloning, and that's how we are going to design primers. So this slide simply talks about how to fetch the sequence of the gene that you want to use as a template for your cloning experiment. And we all are very much aware about the NCBI database and how to look for the gene sequence over there. So let's just go to the next slide. So once you enter the name of this gene, which is MET30 in this case, you end up having various organisms being shown for that particular gene. So obviously we select Homo sapiens. So the name of this gene is mediator complex subunit 30 in Homo sapiens. And this is the information which has been displayed for this particular gene. So if you see here carefully, we choose the transcript variant 1, which is the longest isoform for the particular transcript from this particular gene for our sequence. That redirects us to this particular page wherein you have various features of the gene, where we have the exons, introns, miscellaneous features, and one important thing, which is the CDS, the coding sequence, which I was talking about in the complementary DNA slide previously. So we'll have to essentially select select the CDS region of this gene. Now, once you click on CDS for this particular gene, it has automatically given you the entire coding sequence of this gene, beginning with this particular, if you see the start codon, and this TAA, which is the stop codon, and these intervening uh, regions, all the introns are not here. First of all, a point to note, and these are the 5' and 3' UTRs, untranslated regions of the gene. So we are only interested in taking up this particular sequence, beginning from the start codon and ending in here at the stop codon. So once we have taken the CDS region from NCBI database, we simply have to open this software known as Gene Runner. And it's widely available across various platforms, operating systems, and paste it. Now you see that it is displaying you both the strands, the sense as well as the antisense strand. Though we had only taken the sequence which was by default the sequence of the sense strand reading from 5' to 3' using NCBI software. So it's very good when you'll have to design the reverse primer in this case, because you wouldn't have to essentially sit and reverse complement the sense strand sequence. So that's how this software looks, and essentially you can see that the sequence is beginning with the start codon as ATG and ending with the stop codon as TAA, and that's how the software looks. Now, what you can essentially see here is that we are trying to design the forward primer for this particular sequence. So as I had told you, the forward primer is nothing but exactly the sequence from the plus strand, which would be variable in length depending upon various parameters, which now we would be discussing in this particular slide. For now, we have taken some 25 bases beginning from the start codon, and then we click on a button known as oligo analysis, which is given shaded in a red box here. If we click over that tool, it will open up the sequence of the particular oligo that we have chosen, and it will display you various parameters. So you can see that it is displaying you the propensity of this sequence and its parameters pertaining to formation of secondary structures and hairpin loops, dimers, bulge loops, internal loops, match sites, etc. Also, very important feature is that it is displaying you the theoretical TM of this particular primer. So we know that the melting temperature for any primer or for a set of primers should vary in a range from 50 to maybe we can say 65 degrees Celsius. And within the two set of primers, the melting temperature should not be greater than 3 or maximum 4 or 5 degrees Celsius. And we all know that PCRs run at very high temperatures of denaturation, annealing, and extension, which are as high as 95, followed by the annealing step, which varies as per the primers that you are choosing, and the extension, which is again at a very high, runs at a very high temperature, close to 72 to 73 degrees Celsius. So there's a very important need to first look into the formation of hairpin loops, if there are any chances for hairpin loops to form. So let me just tell you what essentially are hairpin loops. If you take a particular sequence of DNA molecule and you just see that within this particular single stranded sequence, there are regions which are self-complementary to each other. So it will just bend on itself to form a hairpin-like structure. Most of us have already read about all these things previously in our molecular biology classes, but still I'll explain this. So as you can see here in this particular primer in the dialog box, and I've shaded it as marked that in a red rectangle, you can see the hairpin loop and the TM of the hairpin loop, which is being shown at 45.8 degrees Celsius. Now, the key here is to ensure that the TM of such secondary structures is kept at the minimal level. Why? Because we all know that TM is nothing but the melting temperature, and it's the temperature at which half of the bases are denatured and the sequence gets opened up or destroyed, we can say in false terms. So if you keep the TM as being displayed here as 45.8, and bear in mind that your sequence um, is this for the particular primer, and your PCR would run at around close to 50 to 65 in the annealing step. 45.8 is very close to those conditions, and it's quite a chance, in fact, a very high chance that at close to 46 degrees Celsius and your PCR being run at closer temperatures, temperatures close to 46 degrees Celsius, you'll have stability of this hairpin loop. So what will happen is this particular sequence of primers will just bend on itself to form a hairpin loop, and it will not get annealed to your template. That's again a problem, and it will hamper with the extension even forget extension, even the annealing step would just fail, and it will be very difficult to anneal these primers to your such kind of primers to your template. And hence we choose the sequence of primer in a manner which ensures that the TM is kept at very low values, typically below 25 degrees Celsius is also good enough, just to ensure that this particular sequence of hairpin loops is just destroyed or is just highly unstable at PCR condition, and hence your primer remains, we can say, linear to the template, and there would be no issues in annealing the primer to the template. So now we'll need to have some modifications in this particular sequence. This is essentially that we cannot take here. So let's just go to the next slide and see what best we can work out of this. If you notice here that we have just reduced the number of bases to 19 from the earlier number of 25. And now if you see the dialog box and polygon analysis tool for the forward primer, you see that the formation of hairpin loops is not being shown. So I think we are good to go for this particular forward primer here. And we can see the percent GC content ones, which is close to 50%, and we know that for any primer that the percent GC content varies between 40 to even 60%. And if you look at the theoretical TM values, they are close to 59 degrees Celsius, which is also a good number. So we have to keep in mind that whenever we design the reverse primer for this particular template, now that we have chosen the TM for this forward primer close to 59, our reverse primer TM should be within a range of plus minus 4 degrees Celsius of this particular forward primer. So we can see that we have essentially chosen the sequence of our forward primer spanning from 5 prime to 3 prime, beginning right with the start codon on the sense strand and ending somewhere 19 bases down, and we don't have any secondary structures. And then a TM value, we have made a note of that TM value, and we know the percent GC content as well. So let's just see how we can design the reverse primer for this particular sequence. So now what we are doing is we are trying to do. Now what you have to do is you have to go to the 3 prime end of the sense strand, which is towards the end here. And now since GeneRunner gives you the facility of choosing from the minus strand as well, I mean copying and pasting the sequence for your oligosynthesis, it becomes very easy. Note that I have excluded the stop codon from here. The TAA, which has a reverse complement, namely TTA, has been excluded in designing the reverse primer. So this has been done intentionally. Why? Because in the previous slide where I had shown you the EGFP construct, our gene of interest, which is MET30, is upstream to the EGFP molecule. So now I don't want to introduce a stop codon here, because had I done that, the translation would just stop at this particular stage and your EGFP molecule wouldn't have been translated. So that is not what we want. We want EGFP to express as and when MET30 is expressed inside the cells, so that we can track the localization of this particular protein. Also, we can see whether the transfection has occurred or not. So that's the aim of the experiment. So keeping this in mind, you can see that the stop codon has been removed. Removed in the sense, I have not considered the stop codon while designing the reverse primer. So this reverse primer seems to have a TM of 58.5, which is very close to what we had designed for our, which we had obtained for our forward primer. And you can see that hairpin loops may be formed, but the TM is very low, which is not a case of worry. And I think we can just simply go ahead designing this particular primer, reverse primer, for the CDS region of this MET30 gene. And very important thing to note here is that we have excluded the stop codon. The TAA towards the end has been excluded. Had I not taken the EGFP construct, I would have just cloned this particular gene in any particular vector. I would have taken the stop codon. But in this case, since we want EGFP to express as and when MET30 is expressed together as a fusion product, I don't want a stop codon in between. That is why from the reverse primer it has been excluded. Let us discuss about EGFP. So in our lab previously, this EGFP sequence, it was cloned into pcDNA 3.1 vector backbone, which is primarily used in transfection experiments in our setup. So what we had done was we had taken out the sequence of EGFP from this particular construct, as you can see here, and this EGFP sequence is being shown here in green, as you can see. This sequence was excised from this construct, from the particular using the correct restriction sites. And from that sequence, the primers were designed so as to clone EGFP into the PCDNA 3.1 construct, wherein you have to put your MET30 gene as an insert so as to obtain a fusion product. So that is why I have shown this to you briefly. And now we'll see how the primers for EGFP would have been designed. So we'll have to now design the forward primers for EGFP as well. Now for that, we have excluded the start codon, as I had told you previously, based on the construct which I had depicted in the previous slides, that we need to have a fusion product, and hence we should have the MET30 gene, and EGFP should have been cloned in frame with that. So now once the translational machinery of the cell gets active, it will simply make the entire protein at once, and there would be no intervening stop codon from the MET30 gene and an intervening start codon for this particular EGFP molecule, which comes right next after the MET30 gene. And that is why EGFP sequence we have taken, and but we have removed the start codon while designing the forward primers. So you can see for the forward primers, your TM value is close to 27.9 for the hairpin loops, but still this can be managed. There are certain instances wherein we have to compromise with the primer length as well, but it's okay so long as the PCR works, because 27.9 wouldn't be a problem again, because you are doing annealing at temperatures as low as 55 and not going below that. So ideally it should work, and this particular hairpin loop, if at all it exists, should be very unstable at that particular temperature as high as 55 or 60, as in when the annealing occurs. So that's how you have designed the forward primer for EGFP. Now coming to the reverse primer, carefully note that we have now incorporated the stop codon, because now we want the translation to get stopped, because EGFP is lying downstream to the MET30 insert. So we need to incorporate the stop codon while designing these primers. Hence, stop codon has been included, and now you can see the primer which has been designed is not having any hairpin structures, and the TM values, if you note carefully, for both the forward and reverse primers for EGFP are comparable within a range of 3 to 4 degrees Celsius. That is essentially what we wanted. So now we have the primers for EGFP as well. Now once we have designed the primers, and now we want to perform cloning into a particular vector backbone, it is very essential to know about the particular vector backbone itself. To your left in this particular slide, PCDNA 3.1 has been depicted, and you can see various elements of any eukaryotic transcription and translational machinery being present here. And then you have a multiple cloning site, and then you have antibiotic resistance gene, and we more or less we are aware about features of various mammalian constructs. And to your right is essentially a schematic of how the fusion product or the fusion protein would be made, but that would be made only when you have certain modifications in PCDNA 3.1, which is depicted to your left. Now, if you see in the modified vector, which is there towards the right, you have an MCS which has restriction sites for various enzymes, which are HIN3, BAMH1, EcoR1, NHE1, so on and so forth. So the idea is to choose restriction sites smartly in a manner which generates sticky ends in the vector. So generation of sticky ends would essentially be very important, because when you take the vector and you take the PCR amplified product, being MET30 gene and EGFP in your case, together, the common restriction sites that these particular genes would share with the vector would enable generating the same kind of sticky ends. Say, suppose you want to take up HIN3 as the restriction enzyme, and you want to incorporate HIN3 restriction site even in your insert, you will have to modify the primers accordingly. But let us not go into that. That we'll deal with later on. First step is to identify which restriction sites are to be taken for generating the sticky end. So a similar restriction enzyme, the same restriction enzyme in any two different DNA sequences would generate the same kinds of sticky ends. So these would just ligate, and hence you will have a perfect clone. So that's why this particular image has been depicted here, so as we can appreciate why particular restriction enzymes had been chosen in the vector backbone, and how these particular restriction sites were incorporated in the insert. So that is something that we are going to see in the slides to come. Depicted here has been a tool from New England Biolabs known as NEB Cutter. So if you just open that tool and paste the DNA sequence, the CDS of MET30 gene in this case, it will display you a map of the restriction sites which are there on this particular MET30 gene sequence. One careful thing to note here is that before choosing restriction sites for cloning in the vector as well as in the insert, we have to make sure that none of the restriction sites which are available in the vector are present anywhere in the intervening regions of the insert. Because if that happens, now you digest with that particular enzyme, say suppose for which you have restriction sites, say suppose in the MET30 insert as well in the middle of the MET30 insert and in the vector backbone, your MET30 DNA sequence would also get broken down into two pieces. That is what something we want to avoid, because we want to begin the cloning right from the ATG, that is the start codon. So we have to ensure that none of those restriction sites are present here in the intervening regions of this particular gene. And so that is why we have to make a smart choice in choosing the restriction site. The same holds true for eGFP as well. We have to avoid any restriction sites which would be present in the intervening regions from this eGFP DNA sequence, and hence we'll have to uh, choose the restriction sites very wisely there in the vector backbone, because we don't want eGFP molecule to get chopped off in the middle while cloning it. So that is why a careful choice has to be made. So this is one tool by Thermo Fisher Scientific known as Double Digest Calculator. I'm sure there are other tools as well from various other companies, wherein you have to make a choice of your restriction enzymes that you have taken for cloning. You have to simply enter their names, and then it will give you certain parameters whether the two enzymes are compatible. If yes, then in which buffer they are compatible, because compatibility here would mean that you are going to perform cloning in a single solution, in a single vial. You have to add two restriction enzymes there. So there must be a buffer which supports the activity of both the restriction enzymes altogether. Otherwise, one enzyme would just work, the other one would not work, and vice versa. But we want both of the enzymes to work together while we are doing this particular reaction. Hence, we need to make a careful choice of the buffer. So that is why we have this double digest calculator provided by Thermo Scientific. Now, based on the given schematic, it's pretty evident that we want to choose restriction sites very carefully. So for this particular cloning, we have decided to go ahead with choosing HINT3 and EcoR1 as the restriction sites for MED30 cloning, and EcoR1 and XHO1 restriction sites for EGFP cloning. Now, these two clonings would not be performed all together at once. The reason is that if you clone MET30 and EGFP at the same time into this particular vector backbone, you have a common restriction site, which is EcoR1 here. So then again, that might create problems. And instead of MET30 being cloned upstream to EGFP, there might be a chance, there might be a chance that the sequence of the clone which you obtain might get reversed or get scrambled, is the word to use here, and you might not get the desirable product. So first, either perform EGFP cloning with the vector, or perform MET30 cloning with the vector, followed by the second option. So that has to be kept in mind. So finally, the restriction sites chosen have been HIN3, EcoR1, and XHO1. EcoR1 and XHO1 is the region between which you will have your EGFP being cloned, and HIN3 and EcoR1 would be the region where you'll have MET30 gene being cloned into this particular vector to obtain a fusion product. So once you have decided on the restriction enzymes, the sites to choose for performing your cloning and the double digestion, you'll have to see the reaction conditions using the NAV double digest calculator tool. So for that, you just enter the name of the restriction enzymes. In this case, we have HIN3 and EcoR1 for MET30 cloning, and we have EcoR1 and XHO1 for EGFP cloning. Let us look at first the Hin3 eco-R1 pair. So you see the incubation temperature is being shown as 37 degrees Celsius. So 37 is when both of them would have an optimal activity. So that is certain. That is certain now. And then you see a panel of buffers which have been shown here, and they have also given the restriction enzyme activity as a percentage in these particular buffers. So you can see you have blue, green, orange, and red buffers, and there is a common buffer known as tango buffer supplied as 1x or 2x in the concentration. So one point to note here is that both the enzymes should be compatible in a particular buffer system. So make the choice of the buffer system in a manner that both the enzymes work. So for the Hin3 equivalent pair, tango buffer at 2x concentration could be used, because if you use tango at 1x, you can see that eco-R1 doesn't have any activity. And in other buffers, you see wherever NR has been written, it means there is no restriction enzyme activity. And then R pair has to be avoided, though it has 100% activity for both the enzymes, because in R buffer, eco-R1 seems to have a star activity. We all are aware about the star activity of restriction enzymes, and that essentially leads to some non-specific cuts, or maybe the products which are generated have different sequences, so that might create a problem while performing cloning. So we go with the tango buffer at 2x concentration. Now if we see the eco-R1 XHO1 pair, a similar panel has been reported, and you can again simply go ahead with the tango buffer at 2x concentration, because there is no star activity, and both the enzymes seem to show 100% activity in tango buffer at 2x concentration. So all these conditions have actually been optimized by the company, and that is why they have made this particular database and this tool, which comes in very handy. You just have to enter the name of the restriction enzymes. You can just choose the buffer system and also get the incubation conditions as well, which are given here. So that makes our job very easy. So far, what we have done is we have designed primers for MED30 and EGFP cloning based on the start codons and stop codons accordingly. And then we went checking for the restriction sites and doing a double digest and trying to get the conditions for performing the double digest. Now, what is important is the vector has got the restriction sites, but the insert doesn't have, because we have just designed primers based on the CDS region. And we knew that the CDS region, within the CDS region of all the genes, while choosing a restriction site, we have to avoid those particular restriction sites there, as we are choosing from the vector, because they will get chopped off from the middle. So that is why we have avoided it. So what we can do is we can simply add the sequences of the restriction sites in our primers. So that what that will do is it will just incorporate the restriction sites, making the sticky end digestion and ligation very simple, without any worry about getting these sequences chopped off in the middle uh, due to any of the restriction sites being present in the CDS regions of these genes. So as you can see here, we are simply adding the restriction sites. Based on the vector map which I had shown you, it was planned that MED30 would be uh, cloned in the region upstream to EGFP. So if you can see, I have made color codes, and you can simply just have a look at those. So there's a sequence for Hint3 which has been put in the primer, that is AAGC and start codon, as you can see, has been put up, and it was originally there in the forward primer itself for MED30. Now, one thing to note here is that we have also incorporated the Kozak consensus sequence. We all know the importance of Kozak sequences in eukaryotic translational machinery. So that sequence must be incorporated as a mark for beginning of the translation. And once you begin translation, you encounter the start codon, which we all essentially want. So that establishes the open reading frame. So that is essentially the sequence of forward primer for MED30, having the restriction site and the Kozak consensus sequence. If you look at the reverse primer sequence, which was taken from Gene Runner, has been shown in black color. And what we have additionally added here is the restriction site for eco-R1, because we are planning to clone MED30 in between Hint3 and eco-R1 restriction sites in the particular vector. So that has been added. Going to EGFP, the forward primer sequence has been taken from Gene Runner, shown in red, and we know that it is going to span from eco-R1 to XHO1, so the forward primer must have restriction site for eco-R1 being put up here. Now, when we talk about the reverse primer, we must have the sequence of the restriction enzyme being XHO1 in the reverse primer at the 5' end, and we all know that we had designed primers keeping in mind the stop codon for EGFP. I mean the stop codon, which would just stop the translation of the entire fusion product at once. So that's pretty much about how you can add restriction sites and incorporate the Kozak consensus sequence from where you want to begin the translational machinery, keeping the ATG in mind and incorporation of the stop codon, which has been explained previously. Now, once that we are done adding restriction sites in our primers, the only job that remains is adding overhangs to the primers. The reason why we add overhangs is that enzymes, we all know, are proteins, and proteins are made up of their primary, secondary, and tertiary structures. So when you provide any solution containing enzymes under the particular chemical composition and temperature conditions, those enzymes would exhibit a tertiary structure. Now, there might be a possibility that the tertiary structure of the protein or the restriction enzyme in this case might not essentially recognize its restriction site optimally. So, we are facilitating for the optimal recognition of the restriction site by the enzyme in the sequence that we are supplying for the digestion. Hence, we are just adding these adenines and thymines as nucleotides in the overhangs, varying from 4 to 5 nucleotides in length, so as to facilitate the recognition, perfect recognition of the restriction sites by the restriction endonucleases. Okay, so far what we have discussed is that we have designed primers for the construct. We performed cloning for EGFP. Say we performed cloning for EGFP before. We had obtained a clone which had EGFP in it. Now what we did was we took MET30, the amplified product, and we ligated that into the vector backbone, and we got MET30 also ligated all together. So how does that look then? Then this is a description, as you can see, taken from Snapgene viewer at the top panel, and you see the entire product. You can see that EGFP is spanning right after the insert, which is MET30 in our case. And you can carefully note that the start codon begins from the MET30 towards the left, and the stop codon occurs in the end of EGFP. That is essentially what we wanted. I have also made it easier for you to understand as to how this particular clone would be in terms of the DNA sequence. So if you see the fusion product in terms of the DNA sequence, you can see in orange the beginning, the primer, forward primer region for MET30. And yes, you have a start codon, then you have the entire MET30 being spanned. And then there was to be a stop codon in MET30, which we had avoided in designing while designing primers. Hence, that stop codon was removed. And then from GTG in red, you have the beginning of EGFP without its start codon until the TAA, which is the stop codon for EGFP, where uh, the entire translation would halt, and hence we'll have the fusion product right from the start until the stop, having MET30 upstream immediately. It will have EGFP. And now if you just take this particular construct which has both of these together and EGFP as a reporter molecule, which is cloned in frame with MET30, which is our insert, we just have to transfect this in cells. And if you remember the previous lectures on EGFP transfection, you will obtain some images which will be the fluorescent signal coming from the reporter molecule being EGFP here. Now, one thing which I wanted to say while concluding this lecture is that we were talking about GFP. Then now we are talking about EGFP. EGFP is nothing but just a variant of GFP. It is called enhanced GFP. There have been certain modifications which were made in the sequence of GFP molecule to render it more compatible in terms of its the quantum yield and the fluorescence efficiency and various spectral parameters, as to say. So that is why EGFP was taken and is being very commonly used as a reporter molecule in various applications. So this brings us to the end of this lecture. So thank you very much for attending this lecture. And if you have any doubts, feel free to email me at this particular email ID. I'll be happy to answer all the doubts that you have over email. Thank you once again.
Yeah, so we're also done with lecture 2 now. So if any of you have any doubts, any queries, any questions, please feel free to ask. Any doubts? Please ask me if there are any questions, any doubts. Okay then, so I'll stop the recording. Can any one of you confirm that I'll just stop the recording now and just confirm me whether it has been stopped or not.